Hi everyone, welcome to this M2D2 talk. Our speaker today is Pranam Chatterjee. He obtained his PhD from MIT, the Media Lab, where he developed robust genome editing technology that represents some of the broadest, safest, and the most effective CRISPR enzyme to date. Currently, Pranam is assistant professor at Duke University, where his long-term research goal is to design de novo protein-based therapeutics by integrating the newest advance in artificial intelligence with robust experimental engineering platforms. Today, he will be talking about his recent work about designing structure-independent peptide binders via generative language models. Thank you so much, Pranam, for coming here today to share your work with us, and looking forward to it.
Wonderful. Thank you, guys. So hopefully everyone can hear me very well. First of all, thank you, Prudencio, for the very kind introduction. And thank you to all of you for being here and spending part of your day listening to our work. So my name is Pranam, as Prudencio mentioned, and I'm a new professor, actually a 1 year in to being a professor at Duke University in biomedical engineering and computer science. And today I'm really excited to share with you the work that my team has done to develop structure-independent peptide binder design algorithms using generative language models. Before that, I'm going to give you a little background on my group. So my group integrates two very powerful platform technologies, one being algorithms and the second being proteins. You see, we develop very diverse algorithmic modalities, from machine learning methods, which we'll talk about today, bioinformatics algorithms, graph theory algorithms, and structure-based modeling to discover, characterize, and engineer new and useful proteins. But not just any type of proteins. We build what we call programmable proteins, or proteins that can perform bioengineering applications in a very user-defined way. For example, being able to use proteins to edit DNA at whatever loci you want, or using peptides to bind other proteins and be able to chemically modify them, as well as proteins that can create new cell states, essentially new cell types that the user wants to make. So today we're going to focus primarily on the ability to bind and edit proteins using our generative algorithms. But I wanted to sample a little bit about the work that we've done in genome editing as well as cell engineering. And so we're going to go through both DNA and cell and tissue engineering with our programmable platforms. So we'll start with DNA. And you probably have all heard of genome editing, right? It's the ability to make precise changes at the DNA level to genomes. And this is a very powerful technology. Obviously, you can do a lot if you can edit DNA. One, you can fix mutations that cause specific genetic diseases, or engineer the genomes of crops for improved nutrient content. You can even engineer the genomes of microorganisms and crops for improved biofuel production. And genome editing has already been used to shape our ecosystem with tools like gene drives, where you can engineer the genomes of organisms until all organisms in a population have a specific trait. For example, malaria resistance in mosquitoes. But most of all, genome editing has already accelerated the science we do today. It doesn't take years to make a mouse model to study a specific disease. Because we have the ability to edit DNA so precisely, we can do that in a matter of months. And these are just a few of the reasons why genome editing is such a powerful technology. And we have some amazing gene editing tools, none better than CRISPR, which I'm sure you've all heard about. You see, though, CRISPR is not something that we as genome engineers invented. It's actually something that nature gave us. It's a bacterial immune system. Bacteria use CRISPR to protect themselves against viruses. And so we, as a community, have adopted this gene editing, this DNA cutting system to be able to edit the genomes of plants, of animals, of humans. And this is how it works. It essentially requires two parts. One being an RNA molecule that can guide an enzyme, the second part, a Cas9 enzyme, to a specific target sequence and be able to cut it and edit it. But this is a natural system. This is something that nature did not intend to be used for genome editing, and so it has its limitations. And the one that we have focused on a lot in our lab is this small requirement for a sequence beside the target. So essentially, you need this PAM sequence beside the target sequence for this Cas9 enzyme to be able to bind and to edit, right? And if you take a look at the commonly used CRISPR enzymes, you'll see that the one that almost everyone uses requires 2 Gs beside the target sequence, N being any base. And so if you do the math, that actually limits you to 1/4, there's 4 bases, to the second 2 Gs, times 2 because you can cut either strand. So liberally speaking, you can edit only up to 12.5% of DNA using the current set of CRISPR enzymes. And so what we've done over the last few years is engineer new CRISPR enzymes to be able to edit more DNA sequences. And to figure out how to get those enzymes, we have actually developed a comprehensive suite of bioinformatics algorithms called the search for PAMs by alignment of targets, or SPAMalot. You see, SPAMalot can easily and very efficiently use string search algorithms to align bacterial DNA sequences to viral DNA, which contains the invader information that allows us to effectively predict the PAMs of Cas9s that have already been identified and characterized experimentally, but also allows us to find Cas9s that had never been discovered before. You can see the PAM sequences are very distinct. And so what we've done over the last few years is engineer broad targeting Cas9 enzymes using these algorithms, both experimentally. We've engineered a Cas9 called SC-Cas9 that only needs 1 G, not 2. So you can edit up to 50% of all DNA sequences. We've also been able to create a very high activity and low off targeting enzyme. So this is a very safe enzyme that only needs a single G. So it's a very optimized CRISPR enzyme. And finally, we've even extended our enzymes targeting capabilities to target A's rather than G's, which is a very new finding. So iSpy-Mac is able to do that. So feel free to take a read and use some of our enzymes if you're doing any genome editing. But why is this useful, right? So take a look at a gene that you may want to fix. This is a disease allele from Duchenne's muscular dystrophy. And you can see if you use the original CRISPR enzyme, the original Cas9, you're not able to edit very much of the DNA. But if you add in our optimized Cas9s like SC++ and iSpy-Mac, now you can see that you have much more control, much more programmability on where on the DNA you can actually edit. And that's the power of our enzymes. You don't have to take my word for it. You can take a look at all the work around the world, all the groups around the world that have used our enzymes to engineer various organisms' genomes, from rabbits to rice to wheat to even mutant human embryos. Our enzymes are very widely used for various genome editing applications. But while we were doing all of that, right, and it's even actually very recently been used, our enzymes have been used to rescue spinal muscular atrophy in vivo. So we're really excited to see our enzymes be translated. But look, while we were doing all of that, one of my very close collaborators, Ben Kleinstiver, engineered a near PAMless CRISPR enzyme. So this Cas9 only needs an A or a G at the second position, and it's very quite broad, so it's called SPRI. So the question that we've asked in our lab is, can we actually get to a fully PAMless, no PAM Cas9? It has complete 100% targetability of DNA. And so what we've done is we've used structural modeling and combined SPRI and SC++, which is one of our enzymes, together to create a chimeric enzyme called SPRI-C. And as you can see, SPRI-C we've shown to be able to edit at all the different PAM sequences. So we really have not found a specific PAM sequence that we cannot edit. We're able to be very broad in our DNA sequence targeting. And we've shown that our editing is not only very efficient, it edits very well, but we also show that this enzyme has lower off-target effects than SPRI and can mediate therapeutically relevant edits, meaning we can edit at disease-related loci. This is the Huntington locus, which causes genetic diseases. And so while we've done all of that, as you can imagine, over the last few years, we've collected a lot of PAM data, which we've curated into very specific and strict PAM data sets that map a Cas9 amino acid sequence to a known PAM. And basically, we are now training contrastive learning algorithms that can learn the relationship between the amino acids and the PAMs in sequence space. The goal here is not to create broad targeting Cas9s, but rather to create Cas9s that only target one PAM and one PAM only. And why would you do that? Why would you want to create a catalog of hyper-specific Cas9s? And that's because if you can target, say, AAA, but not, say, the other PAMs, you can do what we call allele-specific editing, where if the disease is caused by one copy of a gene, then you can only target that copy, only cut that copy, but leave the other one intact. This is a very powerful therapeutic platform, and we're really excited to apply our allele-specific Cas9s in the future to various different somatic autosomal dominant diseases. So hopefully I showed you a very nice overview of our capabilities of engineering new DNA targeting CRISPR enzymes. And I want to quickly go to the cells and tissue side of things before we really focus on the proteins. So one of the most exciting classes of proteins that you can engineer for therapeutic applications are transcription factors. These are proteins that can essentially turn on and off the expression of different genes. For example, RUNX1 can turn on all of these different genes. But not only can they regulate other genes, they can actually regulate each other as well. So transcription factors create what we call large gene regulatory networks that define a specific cell state. And so people have taken advantage of this. You can take a starting cell type and you can actually throw in different transcription factors and reprogram that cell to another cell type. You may have heard of inducible pluripotent stem cells. That won the Nobel Prize a few years ago. Essentially, it's very simple. You take 4 transcription factors, you throw it into skin cells, and you make stem cells. And people have figured out, other groups have figured out that if you throw in transcription factors into stem cells, now you can make all the different cell types. And so a lot of groups have taken very didactic and step-by-step approach to identify the combinations of transcription factors that make different cell types. So that's pretty cool. But the question that I asked a few years ago was, hey, could we make the hardest cell type known? And that is an oocyte, which is a germ cell. That's the most difficult cell type to make because oocytes are very finicky cell types. They're globally demethylated. They undergo meiosis. They're not your normal heart cell or skin cell. And so the question we want to answer is, what are the sufficient set of transcription factors that we can use to make oocytes? But first, how can we do this in a systematic way, in a computationally efficient way, so that we're just not testing random combinations? And so to do this, we've created an integrated transcription factor discovery toolkit where we start by using graph theory algorithms that can essentially learn the gene regulatory network of specific cell differentiation processes, for example, that of oocytes. So we get a graph representation of the process, and then we apply centrality algorithms on top of that GRN inference to be able to find the most important transcription factors, the most centrally connected nodes in the graph. And we found that if we do just that very simple 2-step process, we're able to identify effectively the transcription factors that make all the different cell types that we want: neural stem cells, myoblasts, melanocytes. And what we do, though, we actually experimentally test this. We put these into stem cells and screen them combinatorically. So some cells may have 10, some cells may have 5 of these prioritized factors. And then we conduct single cell sequencing after we induce their expression. So we know that, hey, cell number 1001 had these 5 transcription factors, and thus we're able to create this specific cell type. So this integrated pipeline allows us to make new cell types in a very systematic way. And so what we've done, you can take a look at our paper, and it's called STEM Screen, and it was published recently in Cell Reports Methods. What we have done is identify transcription factors that if you put them in stem cells, will be able to make oogonia-like cells. These are transcription factors like ZNF281, SOLH1, and LHX8, which are overrepresented, right, in cells that have high fluorescence levels indicating oogonia specifications. These cells are almost oocytes, as I would say, right? So how do we improve this process? Well, we found that if we just start combining these transcription factors together and add in other factors that are important in the gene regulatory network, we're able to identify factors, as you can see here, this D3, DNR3, all 9 transcription factors that make very stable levels of these oogonia-like cells. So we're almost close to making oocytes from stem cells. And so we've shown that this combination of transcription factors makes cells that look very transcriptomically similar to actual oocytes. So shows you how graph theory can be utilized to make new cell types. And so these are what we call inducible oocyte-like cells. And so we've shown that if we grow these inducible oocyte-like cells in culture, they actually grow and they keep growing, and they actually form specific morphologies that look like actual real oocytes. So pretty cool stuff. With just using proteins, we're able to make very new and very difficult to generate cell types. Can be used for various applications. We've actually put this work out in the bioRxiv if you want to take a look. And we've also used the same process to make ovarian follicles. So actually reconstitute an ovary in a dish so that we can study the biology of the human reproductive system. And so we've done the same process, the same STAM screen method to make functional ovarian cell types. This actually work has led to one of my first companies called Gameto, and we've recently raised over about $80 million almost to the point to push this work forward. So we're really excited to see what the company does with our transcription factor-based engineering platforms. So by the way, the quest to make a human oocyte continues. My postdoc, Lynn, who's here on the call, she's working on actually going from oogonia to oocytes, crossing that last boundary. So we're really excited to use CRISPR-like systems to make that happen. And we also are working on modeling how can we engineer new proteins to make new cell types rather than relying on the natural set of transcription factors. My student Srikar is working on connecting representations, deep learning representations of proteins, as well as deep learning representations of single cells, and combining that in a system that can learn the perturbation effects between proteins and new resultant cell states. So we're really excited to see what that will yield. This is very new stuff in our lab. And so hopefully you've seen a nice summary now of both the DNA and the cell and tissue work that we've been doing. Finally, we're going to focus the rest of today's talk on what's super important, which is the ability to bind and to modulate proteins, right? This is where the drug discovery efforts in our lab really come in. And a lot of that work actually began because of COVID. You guys probably know all the scientific progress that we were inspired by to treat SARS-CoV-2, obviously to the development of vaccines like the Moderna and Pfizer vaccines, as well as monoclonal antibodies, which essentially can bind to, these are proteins that can bind to viral particles outside of the cell and prevent and neutralize those viruses so that they cannot enter the cell and replicate. But the bigger question that we were really thinking about was, what if SARS-CoV-2 still infects the cell? How are you going to prevent it from replicating and infecting further? And so to solve this question, we focused on the interaction that SARS-CoV-2 makes with the human ACE2 receptor via this receptor binding domain interface. And so what we did was we were like, okay, that is the interaction via which SARS-CoV-2 enters the cell. Let's use that structural information, right, of ACE2 and the RBD of the virus to engineer and cut regions from ACE2 that make high affinity interactions with the receptor binding domain. And what we did with Manu and I did in the lab, Manu and I were colleagues at MIT before I came to Duke. What we did was we created a pipeline where we could use both co-folding as well as in silico mutagenesis to optimize the peptide sequence and redock them back to the RBD until we could find peptides with very high affinity to our target. Very straightforward, structure-based ways to cut region peptides from an interacting partner and be able to dock them to see if they bind with high affinity, right? So just a good binder. But look, we're not just wanting to bind to viruses inside of the cell. That doesn't really do anything. You're just holding the virus there. Rather, what we want to do is actually get rid of it, right? And so what we did was we fused this peptide, these peptides that we had engineered to E3 ubiquitin ligase domains, what we call the CHIP E3 ligase. Basically, E3 ligases can ubiquitinate and degrade proteins, and they naturally bind to specific targets, right? They have their natural binding partner. What we did was we were like, okay, let's take out the region of these E3s that bind to their natural partner and replace them with our peptides. So now the E3 ligase can go to different sequences, like in this case, the receptor binding domain protein, it binds there and can then thus facilitate ubiquitin transfer to the viral protein, leading to the degradation of that virus in the proteasome by the ubiquitin proteasomal system. So essentially, you can just degrade viruses or degrade proteins by binding to them and bringing an E3 ligase close to them. And so what we've done is we've conducted viral testing of our system and showed that when we introduce our peptide fused to an E3 ligase domain, we can reduce the infection competency of the virus by over 60%, which is a really exciting tool for potential antiviral applications in the future. But the fact is, we built a peptide that can bind super well to viral proteins. By the way, you can read about our degradation platform in our recent paper, but we've used these peptides actually as diagnostics as well. Basically, we've integrated these peptides as beacons on turf microscopes. Essentially, if the peptide binds, it'll bring two arms in a specific orientation that allows for fluorescence emission and be able to detect that fluorescence using a turf microscope. And that has shown that strategy has been able to identify viral particles with the sensitivity near of a PCR test, but but with the efficiency level of the antigen test. So we're really finding a very nice happy medium between standard viral diagnostic strategies using an engineered peptide beacon on microscopes. And if you want to take a look and read more about that work, we've published that work in Science Advances. So this shows you that these peptides are really powerful. But I want to take you guys back to, I think, the most powerful technology here, which is this ability to degrade proteins, right? We showed that we can degrade SARS-CoV-2 by using these, what we call engineered ubiquibodies. These are the peptide fused to the E3 ligase domain. Can we design a ubiquibody to any target protein, right? That is our goal. Because look, I worked in CRISPR. You saw the ability to edit DNA is now unprecedented. All you have to do is give an algorithm a target DNA sequence. We can design a guide RNA for that sequence. This is for CRISPR. Attach that guide RNA to a Cas9 enzyme, and that Cas9 enzyme can then go bind and edit, right? The guide RNA essentially takes the enzyme to whatever sequence you want. What if we could do the same for protein? What if you could give me a target protein sequence that you want to target? We can make a peptide using an algorithm, attach it instead of to a Cas9, rather to an E3 ligase, and thus be able to bind and degrade a target protein. That would be analogous to how CRISPR edits DNA and would make for a very programmable and very useful system for protein editing. And so how do we achieve this programmability is the main question that my lab has been answering over the last year. And so this is a big problem too. You may be thinking, well, there's other ways to design binders, right? But let's take a look at the about 300 proteins that cannot be targeted by standard small molecule based means. The reason that is because these proteins do not have accessible or cryptic binding sites for small molecules to be able to bind to. No matter how hard you try to find a small molecule, you're just not going to get stable enough binding to mediate a therapeutic effect. And so, and moreover, a lot of these proteins are actually quite disordered. So if you wanted to use, say, an AlphaFold based strategy or a structure based method to look at this protein and be able to model it using a graph neural network or a structure based modality, you're not going to have much luck. For example, you can see a protein like MYC is incredibly disordered, looks literally like spaghetti. And so the question we've asked is, why do we even need structure, right? Like that, it sounds crazy, right? But we believe that we can get around structure to design effective binders to target sequences. And that's a lot due to the inspirations we've been able to gain from natural language processing, which has obviously undergone an incredible revolution in the last few years, right? These transformer-based architectures are able to learn the syntax, the semantics, the grammar, and the over a trillion other parameters of human language by training unsupervised models over tons and tons of text, right? And now you can use that for even generative applications, as you're seeing with GPT. So similarly, as these languages are made up of sentences, words, phrases, and have syntax, semantics, and grammar, protein language models are also able to be trained on large corpuses of protein sequences, right? They're just amino acids, motifs, domains that exist in a protein sequence. And there have been incredible progress by large companies to train just unsupervised protein language models. These protein language models, just from sequence, don't capture syntax, semantics, and grammar per se, but rather structure, function, interaction, properties of the input proteins that we put into these language models. And look, this has gone further into the generative sense, right? I'm sure you guys have seen the explosion of image generation tools, not just language generation tools, tools like DALL-E, which essentially connect a sentence like a corgi playing a flame-throwing trumpet to an actual image of that text. And the way these models work is by learning a joint latent space where the encoding and embedding of a text is associated with the embedding of an image. And the images and text that go close together, like say a cat and a picture of a cat, are close together, while embeddings that are far apart, like a cat and a picture of a spoon, are far apart, right? So this idea of a CLIP model, a contrastive language image pre-training step, has allowed for very robust image generation. And so my student, Kalyan, was like, hey, if you can connect images and text, why not connect peptides and proteins that bind to each other? And so he curated a large data set of known protein peptide pairs and essentially trained joint embedding of peptide and proteins in this CLIP latent space, such that peptides and proteins that bind rank highly with this CLIP score, but peptides and proteins that don't bind rank lowly. And so you can see how that can push the latent space in a manner where we create a very expressive representation of this peptide protein pair. And so this is really effective. We actually found that if we can use natural protein interactions of the target protein, those can be great to cut those interacting protein sequences into region little peptide slices and then throw through our CLIP module. This allows us to say, okay, these are candidate peptides because we know they come from natural interacting partners. Let's see if they bind well to our target protein, right? This would be in green. This would be a protein that binds very well. And so we can rank our peptides, our candidate peptides that we cut from interacting partners with our CLIP module. And we've shown that if we take these peptides and we put them into the ubiquibody architecture experimentally, we can actually mediate degradation of various targets like KRAS or TRIM8, which cause specific cancers. So this really shows you that the ability to do peptide design is very powerful. I see, Prudential, you have a question. I think we'll get questions at the end if you can, unless you want to, maybe something's wrong with my my talk.
No, I think it's a small question by Xingyu. He's asking if it's possible for a binding peptide to have multiple target protein.
Yes, this would be a great question. Xingyu, I'll answer some questions at the end if that's okay. We'll get to your question first. Cool. And hopefully you'll see some of your answers in the next few slides. So I think what you saw is that we were essentially cutting sequences from an interacting partner and throwing them through an algorithm, a CLIP model that could prioritize, hey, does that peptide only bind to this target protein and not others? However, this is just random. We're just randomly cutting and throwing through our CLIP module. Essentially, could we find peptides that are likely to have high affinity to our target protein before screening them through our discriminative model? And so what Garrick in the lab did was leverage the protein-protein interaction databases that we had to identify interacting partners of a target, but then train and fine-tune a protein language model called ESM. We fine-tuned that model with amino acid interaction probabilities. Basically, what is the probability that a specific amino acid makes an interaction with the target protein, where we're able to classify these specific amino acid interaction probabilities and essentially score whether a specific amino acid makes an interaction with a target. And what you can do then is when you have these amino acid interaction probability landscapes, basically, we call this the structure agnostic language transformer module, or SALT. Basically, you can be like, okay, here's a protein I want to target. Let me find a partner sequence for that in a PPI database. Let me throw that through our SALT module that classifies amino acid interaction probabilities, and then look at the landscape of the interaction likelihoods, right, across that interacting partner. You can then just cut those high scoring regions from the interaction partner landscape to identify peptides that should have high binding probability to a target protein. So we call this a peptide prioritization module, or PepPr. And so we call this module SALT and PepPr because we have both the learnable language transformer and the peptide prioritization module together. And we showed that SALT and PepPr, first of all, computationally performs very well at identifying interaction hotspots on interacting partner sequences. So we know that these are the regions of the partner that are making these specific interactions. We also show that we can identify these interaction hotspots on structured targets as compared to other structure-based methods. And we show that this process is actually really fast. So if you want to design a peptide in this manner using AlphaFold, it'll take you about an hour to do so, while SALT and PepPr just using language models takes about a minute. But more importantly, we wanted to test if this works in the lab. And so Lauren and Vivian essentially took those peptides, put them into our ubiquibody architecture, and they showed across multiple targets, for example, disordered transcription factors that have very bad structures that we don't have good structures for. We can degrade proteins like GATA4. We can target proteins that cause cancer, but are fusion proteins like DNA JB1, PRKACA. We can degrade those very effectively. And finally, even proteins that are drivers of many cancers, like beta-catenin, we're able to degrade those as well using SALT and PepPr derived peptides. Now, this goes to Seniu's question, how does this operate in terms of other proteins in the proteome? First, we showed that if you can degrade beta-catenin, there are a lot of proteins that matter in this process that cause tumorigenic signaling. We showed that if we degrade beta-catenin inside of colorectal cancer cells, that leads to reduction in tumorigenic signaling, as you would expect, right? The actual cancer processes are going down. We showed, by the way, that these peptides are binding to their target, but not other substrates with very high affinity. So these are just very high affinity, low nanomolar binders. But more importantly, this is to Seniu's question, we determined that our algorithms can identify peptides that only target beta-catenin, but leave the other proteins intact. The only one that goes down in our mass spec experiments is beta-catenin, but other proteins like STUB1, which is actually our degrader, and the regular proteome don't get changed, right? STUB1 is overexpressed because we actually put the degrader into the cell. So basically, this shows us so far that on the peptides that we have tested, we have very low off-target effects to other protein targets. We're only identifying peptides that are very specific to our target protein. And so if you want to take a look and read more about our SALT module, where we actually have that a preprint up on that, this paper is currently in review, so you can take a look on it on Research Square. But remember, guys, the big thing that's happening here is that SALT and PepPr and cut-and-clip as well use partner sequences as scaffolds for peptide generation, right? And that's okay. There's about 80% of all proteins have a partner sequence. We can identify them from PPI databases. But what if you don't have a partner sequence? Or maybe the partner sequence does not bind to the target with high affinity. Basically, the question we've asked is, can we design these peptides, these guide peptides, purely de novo, just from the sequence alone, right? We're not going from target structure. We want to go from sequence. So let's go back to CLIP, right? I showed you that CLIP module, which I hope was very, very beneficial to see how we're able to encode both the peptide and protein. But remember, with cut and clip, we had to cut from an interacting partner. So the question really for us to first answer is, how do you first create candidate peptides, brand new peptides that we can feed into our CLIP module to rank? Because you can't just feed in every peptide. There's about 10 to the 26th 20-mer peptides. It's too much to screen all together. And so the first question was, how do we make a ton of very naturalistic peptide candidates? And so Sahas in the lab had a very ingenious idea. Basically, he was like, okay, ESM is a protein language model. It can create very expressive representations of proteins. And so we can sample the ESM latent space for real peptides. Basically, here are a bunch of real peptides, throw them through ESM. We basically get embeddings that are very realistic, that are representative of these peptides. We then apply Gaussian perturbations to those embeddings to get perturbed peptide embeddings that we then decode back into peptide sequences. So we take peptide embeddings, we apply noise to them, we then get out new peptides, and then we run it through our CLIP module, right? And the CLIP module can tell us which peptides are good for a specific target sequence. And so this is, we call the peptide prioritization with CLIP or PepPrCLIP. And so PepPrCLIP, first of all, can generate very naturalistic peptides. We show that if based on the amount of variance we add to our Gaussian perturbations, we can find peptides that have very similar sequences to natural peptides. This is the Hamming distance between a starting peptide and a newly generated peptide based on the amount of variance that we're adding to each amino acid. So we are basically perturbing the sequence. But more importantly, we want to make sure our CLIP module is good, right? The CLIP is what tells us if the peptide binds to the protein. And so to train this CLIP module, we actually started by creating what we call a noisy data set, which considers more weaker interactions. These are interactions that are not very strong, are not close necessarily to the target protein. They may have a buried surface area greater than 50 square angstroms, but they're also considering larger protein interfaces. But we then, for evaluation, we made sure that our CLIP module did really well. We created a very strict data set for evaluation where we filter for only strong interactions. We wanted to see if our CLIP discriminator could pick up on very strong peptide protein interactions. And as you can see, if we train on this noisy data set and then test on the strict data set, right, in our CLIP module, our accuracies are much higher for predicting a peptide within the top 10% of valid candidates, and our binary accuracy on determining whether two pairs, which one actually is the binding pair, is much more improved when we train on the noisy data set and test on the strict. Now, if you just train on the strict data set and tested on a held out strict data set, the performance is lower, telling us that these weaker interactions are important to learn to generalize to more stricter and more critical peptide protein interactions. And so our PepPrCLIP module does really, really well. We show that on known pairs, we have very high CLIP scores, but on mismatched peptide pairs that don't bind, our CLIP scores are centered around more like a negative CLIP score, like a negative 0.01 cosine similarity score. So telling us that our CLIP module does a very good job at prioritizing real interactions. We've shown that if we test these in vitro, right, Laura and Vivian took these peptides, put them in vitro, we've been able to show degradation of very critical proteins like FOXP3, as well as TRIM8. You can see that PepPrCLIP peptides essentially ablate this cancer-causing protein that causes Ewing sarcoma. So we're right now putting this into further in vitro, functional, and in vivo studies. But the thing is, the last thing I'll say is, what about target conditioned peptide generation? Right now, we're just making peptides and screening them through a discriminator, a generator discriminator method. But what about target conditioned peptide generation, where we make a peptide based on a target sequence? This is a great question for an algorithm like GPT, right? GPT can take in a text, and based on the text, it'll create a response that is linked to that text, right? Just like ChatGPT does. So there's actually a protein GPT that is trained on just protein sequences, just sequences by themselves. And so what we've done, what Sophie and Leo in the lab have done, is fine-tuned ProGPT to be able to capture peptide protein interactions by feeding it a protein sequence as a prompt and allowing it to generate de novo peptides. And we've trained this on the entire peptide protein data set, which again is not quite big, not quite large, but we've shown that this model still does very accurate reconstruction of peptides that are expected to bind to the target protein. So our perplexities are quite low compared to an untrained ProGPT-2 model. So essentially we're creating realistic peptides. And we've shown that this PepGPT model that we've created has shown some early results that we're able to degrade proteins like beta-catenin and downregulate functional signaling. So kind of showing some experimental evidence that this is working. We've actually, Leo in the lab has actually extended this beyond peptides and looked to protein-protein interactions because we have a lot more of PPIs. And we've been able to actually generate proteins that are able to bind to the target proteins using a GPT algorithm. Our current next token prediction accuracy, which tells us for creating the right sets of amino acids from a target protein, is around 0.93. And we're seeing a very nice decrease in loss over our training step. So this is the early results, but I wanted to share it with you guys that we're able to create PEP protein binders to target proteins just by using sequence. Again, no experimental evidence yet, but we're on that very, very soon. And finally, though, last thing I want to say is, the GPT model is autoregressive, right? We understand that it's only using a decoder, so it masks future tokens, forcing very unidirectional generation. But could we use a form of discontinuous masking to understand and learn in a bidirectional sense between amino acids, but still do de novo generation? So Leo had a very clever idea, was whether we could take a peptide and a target protein and mask out all the peptide sequence, not doing discontinuous one-by-one masking, and then throw that masked peptide sequence through ESM to predict what amino acids should be filled in. But again, doing it all at once, not in a one-by-one or a two-by-two fashion. And by using a cross entropy loss, we can compare the actual peptide to the generated peptide and be able to see whether they did a good job. And so far, we've seen that PepMLM kind of works. So far, the perplexities of our generated peptides are about the same as our test peptides, the real peptides, with the target protein encoded, but our random peptides that we just created really have much higher perplexities. So basically, we're doing a very good job of creating peptides using this purely unmasking strategy. And so we really do need some experimental validation. And so all of these methods are going into creating degraders for all of these intractable target proteins, salt and pepper, PepPrCLIP, PepMLM, as well as PepGPT. And so I started a company on this called Ubiquitix, which we're actually translating these degraders for these clinically relevant targets to the clinic as fast as possible. But last thing I'll say, I keep saying last thing, but there's a lot of really exciting stuff that the lab is doing. Why just degradation? Why just peptide attached to an E3 ligase? If you can bind to a protein, why not attach it to a kinase for phosphorylation, or a phosphatase to remove phosphate groups, or acetylases or glycosylases? Basically, can we induce other post-translational modifications on proteins? And so what the lab has been working on is attaching target binding peptides to other post-translational modification domains. Basically, Lauren and Vivian have attached ligands for deubiquitinases, which can recruit these deubiquitinase enzymes in the cell and essentially remove ubiquitin. This allows the protein not to get degraded, essentially stabilizes the protein. And what they've shown is that these dubbed fusions, these fusions to dubbed domains, dubbed ligands, can really stabilize proteins. Now you see the bars going up. So now we have more of those proteins because they're not getting degraded. And we show that for beta-catenin, we show that for FOXP3, and we show that this has functional effect in cancer cells. We can stabilize this pathway and increase tumorigenic signaling, which you don't want to do, but just for a proof of concept. So hopefully I've shown you from this whole talk that instead of programmable, in addition to programmable RNA-guided gene editing, which we can do now with CRISPR, right? With guide RNAs and Cas9s, we can do the same for proteins, right? We can have a guide peptide, attach it to some post-translational modification enzyme, domain, or ligand, and be able to chemically modify and edit a protein. So programmable peptide-guided proteome editing. So with that, I really hope that this is informative but a thought-provoking talk, and it's all really thanks to my incredible students who worked on the research that you saw today, my computational team, Garrett, Kalyan, Zahas, who've been with me since I was at MIT and Harvard, as well, their Harvard undergrads, as well as Sophie, Leo, and Speaker, who are newer students who've done some really exciting work that we've shown to you today, as well as my amazing experimentalists, Lynn, Lauren, and Vivian, in addition to a bunch of undergrads who've done really beautiful work to validate the algorithms that you're seeing today. I would love to thank our collaborators, Matt DeLisa at Cornell, who've helped us to validate these peptide design algorithms. He's an extremely expert protein engineer, uses more traditional methods, but has helped us to validate our peptides. And finally, to the funders, the Hartwell Foundation, the Coulter Foundation, that have provided support for this research. So I want to thank all of you, and finally thank you guys, M2D2 and Valence Discovery, for having me here. I know it's probably a little different than the more structure-based or physics-based methods that you're used to seeing, but hopefully this can spur some really exciting collaboration. So thank you guys so much, and looking forward to answering the questions that you guys have.
Thank you so much, Pranam, for the great talk. Yeah, different, but we learn a lot, so we are very grateful for that. There's a lot of questions in the chat. Let's start with those. And if you have any question after that, please just raise your hand and jump in.
Great. I think we can get started with Xin Yu's question. Is it possible for a binding peptide to have multiple target proteins? Yes. And the beauty is that the peptide protein embeddings that we're generating, the hope is that that is, I mean, the actual scoring algorithm is used for this specific application. So peptides and proteins are embedded into the latent space with their targets and their cognate peptides. And so there are peptides that have other target proteins. And so the model is tasked with finding and understanding those interactions. And if they do, then we get the model the CLIP score is scored as such. So the hard part is that that's not how naturally CLIP is meant to work. CLIP is meant to really separate things that are very distinct. So a peptide that only binds to a target protein. So the hope is that in the end, the peptide that we do generate from the model is specific to only the target protein. The way we can absolutely test that, Xin Yu, is to do whole cell mass spec, where we can actually look at the off-target effects. But yeah, that's a great question. Prableen, I think it's a very excellent question. Is like, how is it ensured that the peptide you fuse, the E3/2, does not get degraded itself? It's actually a great question that we have answered. So you actually go to some of these like supplementary slides. So we've actually, let me click through them, but we've actually created peptide. We've actually been able to extend the half-life of these degraders, meaning prevent auto degradation by removing lysines from our peptides as well as from our ubiquitin conjugation domains. So basically the ubiquitin gets attached to lysine residues. So if you remove them and the protein still stays functional, then you can prevent auto ubiquitination. And so we've been able to do that. And actually we notice that we get much better degradation because we're not getting self-degradation, because we don't prevent ubiquitin from being added to the ubiquibody itself. So that's a very good question. Hopefully we've been able to answer it. Yeah, so how large is this noisy and strict data set? Yeah, it's not millions, right? So a lot of this data was generated from the PDB. I don't have the specific number. We're gonna release the database right now, but you really, after filtering, especially on the strict data set, you're gonna get more on the 100,000, 80 to like 100,000 level of size of the data set. So it's not very big. It's a hard question, right? Because we want to contain interactions that may be meaningful, but you don't want to noise out you don't want to add too much noise to the training steps. So we want to keep the data super clean. So can you create millions of data points? Sure, from natural protein-protein interactions, but our data sets are smaller than that. Yeah, how long are the peptide sequences? Thanks, Lauren, for answering that question. They're about 15 to 18 amino acids. Those are the ones we test, largely because that allows us to test them very fast and rapidly. But honestly, our algorithms can go much bigger. Our training set peptides can be really small. We have peptides that are like 4 to 5 amino acids, but we also have amino acid lengths that are up 30 and 40, more like mini proteins as well. So we're assuming, we can test longer sequences, but right now they're much smaller. Yeah, a PepMLM is essentially a BERT, but it's a very unique BERT, right? So the standard BERT CNU would have been just to mask, random 15% of tokens, right, during training, which, and then you just ask to fill it in. But what Leo asked was very unique, right? Leo was like, well, let me see if I can go back to it. What Leo asked was, hey, can I just unmask all of them at once, right? So it's almost autoregressive in a format, but it's still using a bidirectional encoder. So yeah, it is a BERT technically, but the way it's doing the unmasking is slightly different than you would do in a standard model. So yeah, I can pull it back, right? So it is full unmasking, masking and unmasking of the target. And that seems to perform very well for de novo generation. Yeah, how do you know, if you go ahead with the protein targets, how do you know that they do not have lysine on protein surfaces, right? So the protein targets, yeah, we can absolutely see if they do have lysines. Most proteins do because they have to be naturally regulated by their E3 ubiquitin ligase. But you know, the hope is that this process is not, the process is not very deterministic, meaning as long as you bind somewhere on the protein and there's a lysine around, that's going to be enough for degradation. Now, could we target specific motifs on target proteins that are positioned closer to lysines? That's something that we can still work on, but we haven't solved yet. But it's a great question. Some proteins don't, and those are the target proteins that we're not going to be able to degrade. But most proteins do, and that's just natural how proteins are naturally regulated. Immunogenicity is a great question. Yes, you could incorporate other information, maybe in a CLIP module to find low and high immunogenic protein variations. But I think the way, you know, the good thing, one thing to note is that these are not going outside of the cell. These are intracellular degraders. So we deliver them inside of the cell, and they're already packaged in lipid nanoparticles. So they don't have immunogenicity. So once they get into the cell, you're not going to be subject to the same immunogenicity concerns as you would be for a therapeutic, like an antibody or some mini protein that exists outside of the cell. So we are planning to do immunogenicity measurements, but right now we don't have any data on that one. Did you try to compare the probability of protein-protein binding with physical-based models? Yes. And so basically, I mean, we don't really know specifically the terms, the physical terms, say, you know, that say for like a force field function would learn. We have not specifically picked up on those terms as yet, at least we've not seen. But what we have done, if you look at our Salt and Pepper paper, we actually have compared performance of our PPI you know, predictors, of our interface predictors, with actual structure-based methods that use more physics-based modeling to identify interfaces. And we find that we still are able to identify those interfaces, meaning that the model is inherently picking up on these physics-based parameters. But the thing is, these are ESM models, right? ESM has parameter spaces of between 650 million to 15 billion parameters. And so how to extract the physics-based parameters is something that we need to be looking into. So we don't know what kind of physics the model is actually learning. We just know that when we use it for generation, it's accurately finding peptides that bind to target proteins. Yes, it's a great, yes, go ahead.
Sorry.
Go ahead.
For example, are they like, if you can visualize what protein residues are interacting with each other, can you see whether they are van der Waals interactions, you know, driven by hydrophobic interactions, or they are salt bridge, they are hydrogen bonding? These are like predominant contributors to the binding free energy.
It's a very good question. You know, we have not done that deep level. We actually more quantifiably compare the interaction probabilities that our interfaces that we predict have versus methods like Peptide-derived, which are predicting more like essentially just energy terms. But again, these energy terms are correlated to, you know, specific terms within a force field. So can we deconvolute this region, right? And say, hey, you know, this region is contributing to you know, the formation of disulfide bonds or van der Waals interactions. We have to do that. So it's a great question, Shasha. I think that's something we want to do. However, if you want to remember, you know, for us, we are really looking primarily at proteins that are unstructured. So while we do benchmark on structured proteins, we're primarily focused on disordered proteins, which, you know, these more physics-based models would not be able to capture. So, you know, it's kind of a two-way street that we have to.
But the insight will still be tremendous for, you know, the insights about disordered protein binding instead of the normal, right?
Of course. I think so too. I think if we can look and do the benchmarking on structured proteins, which we're doing, understand the physics properties, we would hope that those physics properties, those properties that the model is capturing would be extended to disordered proteins that we don't have structures for. I think that I agree with you. Yeah. Yeah, thank you. Peng, I know you said that, are there comparisons with RF diffusion, right? So right now, RF diffusion's best capability is to model and design proteins that are of about 50 mer and bigger. If you go too small, RF diffusion does not pick up on peptide protein interactions, it's purely on proteins. So we really have not yet, until probably PPI GPT we're going to be the first model where comparing to RF diffusion makes sense, because our only complete de novo protein binder generation modules where we would have very equal comparison between in experimental platforms would be our PPI GPT. I don't think you can, we are about to start doing benchmarking of our PEP GPT as well as of PepMLM. And so maybe we'll try to create smaller binders with RF diffusion, but from feedback that we've gotten from other groups, it seems like RF diffusion does not extend to peptides. It's more for small proteins. Yeah, so Andrew, you asked, would there be benefits to longer peptides? Yes, of course. I think that's a good point. And there are drawbacks for us. We're delivering these into cells. And so shorter peptides makes a lot of sense because we want to be able to package these PEP proteins into lipid nanoparticles. But of course, longer peptides can make more stable interactions. However, they can also make more spurious interactions too. So we're very much exploring mini proteins, 30 mer, 40 mer peptides. We can even go to 100 mer proteins if we wanted to. We're not limited to peptides. We just do that more for the drug properties that we want to optimize later. So yeah, so there are benefits. You'll probably get much more specific and probably higher affinity interactions with larger and longer peptides. Yeah. And Harry, I know, asked, would giving up proteins binding pocket help with the peptide generation? Potentially. So one thing I want Harry, you to think about here is for us, and we have to think about as well, is that we're using, underneath all of our models, we're using ESM, and we're using an existing protein language model embedding, right? And so these language model embeddings have not right now been trained on domains or have not been trained on motifs, right? So if we can somehow annotate a protein language model embedding with, positional information of the binding pocket, it may help. However, remember, unlike small molecules, we don't need the binding pocket, right? We can bind and make stable interactions with surface protein, surface motifs, with disordered domains. We just don't need to go to a specific binding pocket. Small molecules, however, have that very, very specific requirement. And it's actually, I think, personally, is a reason why we have an advantage over small molecules, is because I think I even say it here, right in, or actually this is not it, I say it, you're not able to target specific motifs on target proteins if you have to go to binding pockets. Now, could we go to binding pockets? Sure, but if there is a binding pocket, Harry, I would suggest probably going with a small molecule, right? Because small molecules, if there's a good binding pocket, you can stably bind there. But I want to say here, sorry, I'm just going through, these small molecules right here cannot bind like here, right? They have to bind here. Therefore, they cannot discriminate between isoforms of target proteins. And they're also very non-modular and tunable. So I think our platform of using peptides or protein binders gives us a lot more flexibility on how to design these binding proteins. Cool. Does anyone else have questions that you don't want to type, but you just want to ask? I'm happy to answer those too.
I have one last question.
Yes.
Is there any particular class of protein that the whole suite of model have a hard time designing the degrader for?
Say it one more time. Had any difficulty or had? Yeah, it's a great question. Of course, I think the hardest class is our transcription factor fusion proteins. I think it's one of the hardest class of proteins. Our degraders have not done, we've shown you some examples, but I think you can ask our experimentalists that PepPrCLIP or PepGPT or salt and pepper, I think when you go to these oncogenic fusion proteins, when you have two proteins that come together to form one, those are very difficult class. They are completely undruggable with small molecules because they don't have binding pockets. And more importantly, Prudencio, it's very hard to target these proteins very uniquely, meaning only targeting the fusion protein, but not the individual proteins that make it up. That's a very unsolved question. We're going to have to really think outside of the box to do that effectively. But the CLIP module may be the answer to that. I think that's something we're going to explore.
Okay, I see.
Cool. So, sorry, and maybe there's two more questions if that's okay, Prudencio. I know we're a little over time.
Okay.
But Xinyu, I know, asked, how do you think of the future for single cells, like reprogramming? So yeah, it is what we're working on right now, and I think the ability to use proteins, Xinyu, that can regulate cell state, right, single cell states, and push them to new reprogrammed outcomes is really the biggest question. Right now, for these tools, we're just going and degrading one protein, right? But what if you could create proteins, single proteins that can regulate and make brand new large regulatory changes to cells? That I think is the big goal. And I think one of my students, Shrikar, is working on creating proteins that can say, okay, if you want to create an oocyte or a heart cell or a muscle cell, here's a protein you put into a cell, it'll make that new cell type. I think that would be the ultimate dream of cell reprogramming, because then it's very programmable. So yeah, Senior, hopefully that answers your question. But that's what I think. And Harry, I think, is a peptide protein data set public we are about to make. So this PepPrCLIP will be made, is probably, hopefully, fingers crossed, right? We're working on it right now. Will be up on the archive, bioarchive, probably in about a week and a half. So keep out a look for that. And we're going to publish our peptide protein data set alongside of that model. Also, the model will be available as a collab notebook. So you can just press a button and you can get a peptide to any protein you want. And I think my experimentalists can vouch for that it's very easy to use. They're not really coders, so if they can use it, I'm sure you can as well. So yeah, we'll make it all publicly available.